Repair Biotechnologies, a company that's developing a means to reverse atherosclerosis. And I've also been a fairly active self-experimenter for most of the last decade. Done a variety of things, including microbiome interventions. And I present to another self-experimenter group on a fairly regular basis. So I have a bunch of these presentations hanging out on various topics. Good that there's someone who's actually a scientist in the space here to keep me on the straight and narrow. This is very much going to be a presentation focused on the self-experimenter's viewpoint, and the science will not be enormously heavy. But let me see if I can share a screen and get this to happen here, and then I will forge ahead. Feel free to ask questions as we go. Just bear in mind that if something seems obvious, it's probably going to be 2 slides ahead in the future. Can you guys see this? Yes? No?
Yes.
Okay, great. So this is really phrased as an introduction to the gut microbiome in aging. And, you know, if you know a lot about it, I'm probably not going to tell you anything you don't know already. So what is it? So the gut microbiome is the collection of microbial species that hang out in your intestine. It's ever-changing. It's a very complex, dynamic set of competing species. It has some evolved desire to stay the way it was yesterday. So you can push it, you can perturb it in directions, but it will tend to come back. But there are ways to change it permanently. And these microbes do a whole bunch of things. They're really symbiotes. They common cell species. They serve very necessary functions. They enable us to live because of the way in which they process food. Unfortunately, not all of them are beneficial. And certainly it's far from understood, you know, what they all do. There's thousands of species, and some of them are pretty obviously bad, some of them are pretty obviously good, and a lot of them are just question mark. So in the context of the gut microbiome, what does it mean to have an old microbiome? And what this means primarily, from the point of view of the research community, is that the proportions of different species are changing. That you have more bad species that are just running around causing chronic inflammation and or generating metabolites that are harmful, and you have fewer beneficial species that are acting on your tissues in ways that are necessary, such as generating butyrate that induces BDNF expression, that increases neurogenesis, that sort of thing. It's entirely possible that aging of the gut microbiome also involves changes in what these species are actually doing. That's very under-researched. I don't recall seeing a paper on this topic at all. Currently, the assumption is that a species does what it does, and aging is just a matter of you have more or less of them, and that may not be true at all. We've no idea. So this is further complicated by the fact that, you know, you can take a population and there's no one optimal microbiome. Different populations have different balances of these different human populations have different balances of the microbial populations. There are certain things you can say about how things generally go, and researchers are finding all sorts of interesting correlations between specific microbes and specific age-related diseases. But generally speaking, one should not expect anything to be universal here. If you see something happen and is generally acknowledged to be bad, maybe it's bad in 80% of the people. Maybe it's like maybe it occurs in 80% of the people. You know, you're looking at population level studies for a lot of this epidemiology of what the microbiome looks like. So it's entirely possible that all of the findings that are going on right now, these last 5-10 years, where we say, oh wow, this thing is associated, too much of this bacteria is associated with Alzheimer's disease. No, you come back 10 years from now and you figure out that it's somewhat more subtle than that. And we should expect this to progress in that sort of a way, but there are patterns, and it is clearly the case that an aged microbiome is worse than a younger microbiome for enough people to really matter as a point in which you want to make interventions. So there are a lot of these species. I mean, there are a lot, like thousands at the very least. And you can go in there and sequence everything. I mean, genetic technologies are dirt cheap these days. You can hand over a stool sample, you get an exact readout as to what's in there and how many of them there are. You can, by the way, you get like a sequencing of bacterial genomes, these specific genes that vary characteristically between species will give you a complete readout as to what's in there and how many of them there are relative to all the others. But there's just no room to talk about specific species, and I'm not going to, for the aforementioned reasons that they might not be as universally bad or as universally good as people say. And most of the interventions are not targeting specific species, and nor will they, I suspect. So it's not really useful to talk about that unless you're really getting in the weeds. So we're going to talk about more big picture stuff. So, what causes the aging of the gut microbiome? And in this, I mean the change in population, the relative population numbers. So there's a whole bunch of potential candidates. Lifestyle change is one of them. Certainly, if you change your lifestyle for a long period of time, you can expect your gut microbiome to adjust. And older people do tend to exercise less. They tend to put on weight, eat a different diet, and that may be important, but we don't really know to what degree that's important versus the others. Secondly, all the intestinal mucosal barrier becomes less functional, and this is sort of a bidirectional relationship with the gut microbiome. There's stuff going on here that goes both ways. The gut microbiome can be very bad for your intestinal mucosal barrier, but at the same point in time, they like to live in this. Many of the species like to live in this niche. So the important one, for my money, and again, this needs to be proven. But in aging, nobody knows the relative effect sizes of all the mechanisms. If somebody tells you that a mechanism exists in aging, yeah, they're probably right. I mean, 99% of these things turn out to be true. But is it important? How big is it? How big is the effect size relative to other mechanisms? And generally speaking, nobody can tell you that. So, but for my money, immune dysfunction is the important one, because the immune system gardens the gut microbiome. It goes in there and murders microbes that it doesn't like. And one of the interventions that I'll talk about today is just making your immune system do that better. And it's actually very effective. So that is why I think that immune dysfunction is really kind of an important factor in the aging of the gut microbiome, because you can use it to make things much better. So whilst talking about immune dysfunction, we should say that this isn't just one thing. It's not just here's a measure, your immune dysfunction is 5 or 6 out of 10. It's very complicated. The immune system is made up of enormously different subsystems. They do complicated things. Nobody fully understands this. And so there's definitely room here to say, okay, what's actually going on? As for any interaction of the immune system with the rest of our tissues and or common cell microbes, it's a complicated story. So it tends to be the case that healthy older people have better gut microbiomes. This has been demonstrated in a number of studies, and people who survive to very late old age, they tend to have much better gut microbiomes. And to the degree that this is tautological, that's pretty much what you'd expect. Somebody who survives was aging more slowly. Therefore, if you go look at all their systems in their body, they kind of have to be aging more slowly, because otherwise they wouldn't be aging more slowly. It's a big circular logic loop. It doesn't tell us much about, what's the chicken and what's the egg here. Are centenarians, is achieving centenarian status meaningfully influenced by what's in your gut microbiome at age 60 or age 30? And that's an interesting question, and nobody knows. So, or is it that the fact that they have better immune systems? I mean, we know that centenarians have much better immune systems than younger, unhealthy, old people, and maybe that's what's causing. Again, nobody knows. This is an area ripe for people to go spend $100 million and get some better answers. So, moving on, how important is this to your long-term health? So I know I always tell people that you're not gonna get answers as to how important something is. All you can get is suspicions. And the suspicion here, looking at the animal data, is that this is sort of in the same ballpark as diet and exercise in terms of effect size on lifespan, possibly more. It does about the same thing as calorie restriction does if you go rejuvenate the microbiome. So in the example noted here, this is doing a fecal microbiota transplant in tiny little fish. And it restores a youthful microbiome, and the fish are healthier and live longer. So this is interesting. If you have something that's not calorie restriction and does about as well as calorie restriction, maybe we should be looking more into that. because calorie restriction in humans is clearly fairly beneficial, even if you're not going to get an extra 20 years out of it. You're certainly going to get improved health and a few years out of it. So, and this seems to be a equally interesting point of intervention. So, one aside that I'll note is that the gut microbiome changes early in life. Now, things that change early in adult life are very interesting to those of us who are sort of interested in self-experimentation and research and etc. Because these points should be test beds for intervention. Because if you're developing interventions, you don't really want to be testing in 60, 70, 80-year-olds because it's going to be a disaster. It's really hard to not fall into the pitfall of accidentally killing someone, or somebody just dies in your study and that's it, you're toast. So if you can intervene in like 40-year-olds, that's great. Because 40-year-olds, you have a reasonable expectation that even if there are side effects of your therapy, they're going to survive it because they're 40. They're in pretty good shape, relatively speaking. So as it turns out, the gut microbiome does go through characteristic shifts as early as your mid-30s. And why? Nobody knows. This again, the study of aging is generally carried out in old people. So I think we all understand that there are things that clearly happen between 20 and 40. I mean, just look at your skin. But what are those things? Is this just a lower level of all of the normal mechanisms of aging we see in 60-year-olds? Is it some different distribution of these mechanisms? I mean, nobody knows because nobody studies aging in 20 to 40-year-olds. So, but nonetheless, there are things like loss of ovarian function, the involution of the thymus, and changes in the gut microbiome that happen very early, which means that if you're, a 30 to 50-year-old, maybe you can actually go do self-experiments here and see some change. Whereas most of the things that happen to a 60, 70, or 80-year-old, you're not gonna see something. Anecdotally, one of my investors is 80-something, and he's a very energetic self-experimenter, and everything works for him. Everything he's done has an effect size. If you're 80, it's amazing, setting aside the fact that you're 80. But if you're younger, if you're 50 and in fairly good shape, good luck finding a signal out of most of what you can do. But here we have something where maybe we can get a signal. And I'll show you a little bit about that later. So another aside, it's clearly the case that there is a gut-brain axis. And nowadays people are talking about the microbiota gut-brain axis because it's clearly the case that the microbiome is influencing all of this. And as I pointed out earlier, BDNF expression is kind of important. So butyrate is derived from fiber, among other things, and it's one of the reasons why fiber is good for you. One of the many, many reasons. Again, don't ask me about relative effect sizes. Nobody knows. But if the microbiome is very influential on brain function through inflammation, through BDNF, through probably a bunch of other things, nothing ever happens for like 1 or 2 or a dozen reasons in our biology. And nobody can tell you which of the mechanisms are more or less important. But if you want improved brain health, then consider fixing up your microbiome. And that's also something I will mention later. So inflammation. Inflammation really, I mean, wherever you look in aging, there is inflammation. This is the mechanism where you have the most compelling evidence to suggest this is the big important one. It's up there. It's a problem. It's causing disruption to tissue function and everywhere. Unfortunately, you can't. It's hard to address inflammation in aging. All of our current tools for suppressing inflammation are very crude. Think of the therapies for autoimmune conditions. They just sabotage one particular signal, and that screws up your immune system in the course of suppressing the chronic inflammation you're undergoing. I mean, people with rheumatoid arthritis have a 10-year loss of life expectancy, and in large part because of the therapies they're undergoing that remove their immune system's ability to respond properly to normal stimuli. So you have this problem where all the bad inflammation and all the good inflammation goes through exactly the same pathways. So the only way to stop the inflammation is to get rid of the thing that the immune system is reacting to create this inflammation. And in many cases, this is going to be really hard. That's basically the same as saying let's fix aging. Let's remove all of the cell and tissue damage that causes this to happen. But in the case of senescent cells, that's pretty easy. We can get rid of them. In the case of the gut microbiome, yeah, we can totally go get rid of a bunch of these inflammatory bacteria. So, these are good points of intervention, even if they don't solve the whole problem. But I expect that in terms of the effectiveness of microbiome interventions, it's largely a matter of reducing inflammation. That is my suspicion. And obviously, again, nobody's going to be able to tell you whether it's true or not until somebody does a very clever experiment in which they only mitigate the inflammation and do nothing else, which I suspect is next to impossible. So how do you actually determine what's going on in your gut microbiome? And I should say that the fact that you can absolutely just list your microbiome cheaply for like $150 or $100 or something makes it a very attractive target for self-experimentation, because you know exactly what happened. You may not be able to process that data into a useful answer, but you know exactly what happened at some level. So there's a gene, 16S rRNA, that varies in characteristic ways between microbial species. It's very actively evolving, and there's databases that tell you, okay, if it looks this way, what species is this? So you just dump all of the DNA out of a stool sample into your system and just shotgun it and see what you get. And this is now very, very cheap, like all omics technologies. So there are infinite services that will do this very cheaply. So the one I started using back in the day is Viome. They were one of the first. Ubiome was the one that died for bad reasons, but as things moved on, Viome became the cranky old company on the block that really doesn't have as good a product as everybody else, because everybody else came later, had a better chance to build a better thing. So I wouldn't use Viome now. If I was starting now, I would look at some of the others. But basically, there's a bunch of people who have very similar business models, which is you ship them a stool sample, they tell you what's going on, they give you a score of some sort, they interpret the literature for you. And then they will sell you on tailored probiotics, the value of which is dubious. But, pick your poison. At the end of the day, you're going to get some kind of a score, and you can assess before and after very cheaply what actually happened here. So beyond that, if you don't want to talk to these guys, the direct-to-consumer services, there are other groups like core facilities in universities and companies which probably won't talk to you, who will actually provide microbiome service as microbiome sequencing and give you more of the raw data than the services do. But it's tough to find someone who's going to do this at a reasonable cost. Well, there are definitely university core services. I'd focus on those university core facilities where they accept outside people, and you can just give them $100 in a sample and they will just do it. And they give you an output. What you don't get, of course, is the wonderful extra sort of scoring and processing and interpretation of the literature you get with these. Your mileage may vary. And, how much of a scientist are you? What do you want? So you can look at all these things and see, how do I want to do this? But generally speaking, the costs are minimal. Okay, so the interesting part of this question, now that we know that the gut microbiome ages and that's probably bad for you, what can you do to fix this? So every year or so, somebody comes up with something new. This isn't an exhaustive list by any means. But the big two important ones, I think, are fecal microbiota transplantation, where you take a stool sample from a young person and put it into an old person, where a person can be a mouse or a killifish. And this actually produces lasting change. It literally resets the gut microbiome. The new sample outcompetes the old stuff, and you get a youthful distribution of populations again. And that's permanent. It's a permanent change. So flagellin immunization, this flagellin is the component of bacterial flagellae. And to a first approximation, if a bacteria has a flagellae and it's running around trying to get into tissue, you don't want that in your gut. So you can actually provoke your immune system knows that it doesn't want flagellin around, but apparently it needs a reminder. So people have developed flagellin. It's been developed. There's been a clinical trial as a sort of vaccine adjuvant. When you're vaccinating someone rousing up the immune system in a safe way, it helps the vaccine work. So people have tested flagellin inoculation as a sort of vaccine adjuvant strategy, but it hasn't gone very far beyond the first clinical trial, but it does show that it's safe. So you can just inject this into yourself. You can order it, inject it, and your immune system gets all riled up and goes and murders as many bacteria as it can. And this actually does improve the quality of the gut microbiome considerably. Only tested in mice for actual published data. So there's things like icarin and cyclic peptides and a whole bunch of other stuff that are even sort of, very little. There's very little sort of other than mouse data stuff going on there. And not all of these are permanent one-use things. And for my money, I'm not particularly interested in things you have to do on an ongoing basis. That's annoying to me. I'd rather just have a one and done approach that you only have to do once a decade maybe. So obviously one has to talk about probiotics and prebiotics. And if you can use a fecal microbiota transplant to permanently change the gut microbiome, obviously there's some mix of microbes that you could put together hypothetically that you could take in enteric capsules and would achieve much the same result. Obviously that's true, but it doesn't exist yet. So nobody actually does this. All of the stuff you can buy in terms of probiotics and prebiotics, they only have short-term effects. It's questionable as to what the health effect is of these things, and it's really not addressing the problems that people are now discovering. So, but nonetheless, I expect the future of this field to be one in which you do have things that are substituting for a fecal microbiota transplantation, where you're taking ounces or grams of a cultured mix of bacteria in enteric capsules that will permanently alter your gut microbiome. But we're not there yet. I get it. Starting from now, I would imagine a 20-year run if people got really interested in doing this, and to the point at which you have multiple services actually offering this as a thing. But meanwhile, the probiotic and prebiotic industry is quite happy selling stuff that's basically useless. Okay, so I put in a bunch of review papers here. This deck can get sent around afterwards. I'll let the organizers do that, and feel free to run through these and take a look. There's a lot actually out there. It's a very broad and expanding topic these days. And the summary before we get to what I've been doing is, if you care about your health, you should probably pay attention to the gut microbiome. If you care enough to do diet and exercise interventions, then you should probably be doing gut microbiome interventions. Inflammation is probably the primary consequence here. There's a number of ways in which you can rejuvenate the gut microbiome that have really good animal data. There's decent human safety data, and of course, absolutely no human efficacy data to a first approximation. And you can assess the microbiome very cost effectively with any one of a number of services by providing them fecal samples. And that's really the summary of all of that. And does anyone have any questions before I move on to talking about what I did and the results I got? Yes. Just speak up if you do.
Yeah. So this is Chris Wickman. A couple of questions. So I posed some questions before your comment about taking things long term. But what are your thoughts on supplementing with butyrate?
Have you actually ever handled butyrate?
No.
Don't.
Okay.
Butyrate is the reason that vomit smells like vomit. It's not a fun substance to have around.
Okay.
If you're gonna supplement butyrate, don't get powder. Get pills, but you are gonna have to do grams and grams of it. I don't think it's a terrible idea. I think it's a terrible idea to try to have powdered butyrate lying around. It's a miserable substance to work with, and honestly, compared to the other options, it doesn't seem as wonderful because you're only addressing one specific thing. It should be beneficial. Any way to upregulate BDNF should be beneficial, but from my point of view, I'd rather, obviously I have access to things that most people don't, but like I'd rather go run up a batch of AAV and just inject myself with BDNF encoding AAV into a fat pad than go through the trouble of butyrate supplementation forever and always.
Okay. Next question. Any thoughts on what's going on in the gut when somebody is experiencing brain fog?
I mean, inflammation is clearly connected to brain dysfunctions of that nature. If you look at the work on depression, on like a whole bunch of psychosis type stuff, and indeed like post-surgical delirium and et cetera, et cetera, et cetera, it's pretty clearly seems the case that inflammation is an important thing here. So if you can find a way to suppress inflammation, then that's probably good. And I will say that in my interventions, the biggest thing I noticed was clarity of mind. It's night and day, really.
Okay, and a comment on that. Again, this is something that is could be unfortunately a long-term usage thing, but I've had good success with a US-based product called SBI Protect, which is based on serum-derived bovine immunoglobulin. I've definitely noticed less inflammation when I take that. It comes in a powder that I put in my morning shake.
Yeah, not familiar with that, I'm afraid. I can't comment.
Okay, thank you.
Okay, anyone else? Well, let's move on.
I had a question if nobody else asked. So
Yeah, fire away.
One thing was, after you go through like a 7-day or 10-day antibiotic course, what are like some things that you can do apart from like extreme measures like fecal transplant, something, like over the counter, like probiotics? If that doesn't work, what are the other steps that you can take to fix your microbiome?
I mean, there aren't any. That's just, you're kind of stuck there, right? So one of I talked about how the gut microbiome changes with age, right? It changes quite early. One of the possibilities here is that what we're seeing is the effects of antibiotic use on a population scale, obviously intermittent antibiotic use. So that's plausible, but of course, this is a hypothesis. There's no data out there to support this one way or another. But it certainly seems plausible that antibiotics could be, the way we use them in our society today, be causing this sort of a problem. And as to what you can do to fix it, I mean, you can't. Well, that's it. If your antibiotic use changed your gut microbiome permanently, there's nothing you can do to put it back, short of one of the more dramatic things we're talking about here.
That's not very hopeful. I mean, that sounds a bit sad.
Well, I mean, like, knowledge is power, right?
Yeah.
First step to solving the problem is knowing.
Yeah.
I think, like, if you're saying that fecal microbiota transplantation is radical, it's not as much work as you think it is. Understanding the risks is important, but like it's nowhere near as much work to do this as you might think.
I mean, I guess for me, the whole idea of it is a bit eerie. I mean, it's some degree of conditioning to how we perceive it, but yeah, it seems a bit eerie to me because of the idea of it. Like, I'd rather have synthetically produced microbes in the lab, not from another human source.
Yeah, absolutely. It's much better, and I will get to one of the reasons why it's much better, but it's not going to happen anytime soon, unless somebody jumps up and does it.
Cool. Interesting.
Okay. Anyone else, or shall I move on here?
I can see there is a question from Martin in the chat. I don't know if he wants to read it out or if you can see it.
Where is the chat?
The question was with regards of the companies providing or offering the gut microbiome sequencing. But basically you have answered that with Viome and the other companies on the slides. So thank you.
Okay. Yeah, I mean, I've looked at some of the others. I haven't used them because I got stuck. You know, once you start with a specific service, if you're going to do a comparison, you kind of have to stay with it for years, because you can't just switch over to somebody else's metrics, because every company has different metrics. So I've certainly looked at some of the others in summary and thought, well, it would have been great if they existed in 2020. You know. Anyway, any other questions? No. Okay, moving on. So what have I done? I should preface this probably by saying that I have a high risk tolerance, and improving my health is sort of number two after number one is like find things out. So I'm certain, and if you look at like, okay, what happens if I do nothing over the next 40 years? That's not pretty. So what does it much matter if I damage myself a little bit along the way in the course of learning something? And I guess like a number three, motivation was I was kind of irked by the lack of rigor in self-experimentation in the longevity community. I mean, we're nowhere near as bad as the bodybuilders, but you know, people just don't care about effect size. They don't care about testing things. They don't care about writing stuff up. It's so I was kind of irked by that and wanted to at least like show by example that there are better ways forward, which is how I ended up putting a bunch of self-experimentation stuff on Fight Aging as a self-experimentation section there, in which I talk about protocols, how to develop them, how to do the research, how to source therapies, et cetera, et cetera, et cetera. So, but my risk tolerance is not your risk tolerance, and everybody should make the decisions for themselves. So a few notes on if you're going to do a microbiome intervention. To my mind, all of the interesting interventions are the permanent ones. And okay, great, but it's permanent. You can't go back. You're stuck with whatever you got. The only way forward is to do it again with some sort of a different source material. So, and it's certainly the case that the gut microbiome may be generating a whole bunch of interesting medical conditions, and nobody really knows which microbes are doing this or why or how. So nobody can screen for it. So there is an FDA-approved FMT, fecal microbiota transplant therapy, and they haven't addressed any of those concerns, largely because they're treating people with C. difficile infection. And if you wanted to swap out your C. difficile infection for fibromyalgia, people would do that in a heartbeat. And of course, if somebody's had C. difficile and they then have sort of ongoing horrible unpleasantness that's less horrible and unpleasant than C. difficile infection, everybody's just going to say, oh, well, you had C. difficile infection, this is what happens. So I suspect that figuring that out is going to take a while. The measurement we talked about, if you're going to do this, measure what you're doing, use a service that gives scores, use the scores to judge whether you're actually getting somewhere. If you're doing flagellin, you have to have a way to get this from a lab supply company, and they don't ship to consumers. You need a way to look like a lab or have a friendly lab that will receive the material for you, at which point it just goes in a freezer and you learn how to inject things. And injecting things is not as hard as you might think. If you're just doing sub-Q or intramuscular, it's just insulin needles, it's just learning how to make vials. It's not hard. It's just work. Don't do IV injection yourself. Just don't. It's hard. It's like it's something where you really need an extra pair of hands. And there's few therapies are actually really meaningfully interesting on that front. For the flagellin, it doesn't matter where it goes in your body, your immune system's going to react to it. For the fecal microbiota transplantation, you need to source a healthy donor. So maybe you have a 19-year-old friend you can ask, or better, you could use a service. There's this group called Human Microbes that screens donor materials, at least for pathogens. So again, as noted, nobody knows how to screen for things that might cause fibromyalgia or other issues. And then they deliver you the stuff by the mail. And if you're going to do that, then the delivery method is enema. And that's, the literature says do this a couple of times over the course of, some number of weeks, and then see what you get. So my results look like this. This is, you can see I did the flagellin immunization, 10 weeks of once weekly injections of 10 micrograms of flagellin into just sub-Q, and did that in the middle of 2020, and just immediately it made a really big change in metrics that were actually pretty terrible. For me, I was definitely not, my gut microbiome was not in good shape, and after that it was in good shape. So that was great. So then I said, okay, let's do it. Interestingly, you know, that you can see the active microbial diversity sort of trending downwards there as my immune system murders everything it can get its hands on. And then that was restored by doing a fecal microbiota transplant from a young person, which was just a very ad hoc, let's see what happens. Why not? Find someone who's healthy, go do it. In terms of like, why not? If you look at the epidemiology of things like fibromyalgia, muscle lump syndrome, or myofascial pain syndrome, as it's called, there's a whole bunch of things that are sort of in this orbit, poorly designed, poorly sort of categorized things that look kind of autoimmune but don't involve upregulated inflammation and cause generally pain and fatigue and all the rest of it. Nobody really understands how these things are caused. So my donor went on to develop this a year after I did this, and I went on to develop this about a year or two after I did this. So that suggests that you're probably in a 1 to 2% bucket of having problems if you do this sort of thing. Again, CF, what I said earlier about risk tolerance. If I'd known that there was a 1 to 2% chance of something going bad, I probably still would have done this, because we should all be so lucky as to have 98% success rate things in our life to try. Now, interestingly, the FDA approved, there's one FDA approved therapy for fecal microbiota transplant for patients with C. difficile infection. And as I said earlier, you know, if a patient ends up with fibromyalgia, it will be difficult to point the finger at the treatment, but it might be the treatment. So I suspect, as you noted earlier, that the field will move away from this. The fecal microbiota transplantation will never be very widely used, and instead, you know, if not least because it's very hard to make money off this sort of thing, it will be replaced by some sort of synthetic bacterial microbiome that people just assemble and grow in culture, and then, you know, you take it with enteric capsules or enema or whatever works. I think that's going to happen, but this is all sort of, you know, very early days here, and that's what I did. So any questions on that?
I do have a question. Is like, why did you do the FMT at first? Like, did you consider your microbiome, basic microbiome, not healthy? And compared to what?
You're going to have to repeat that. You cut out there.
Oh, apologize. I wonder, like, why did you experiment the FMT at first? Like, did you consider your microbiome unhealthy when you decided to go for it, and compared to what?
Well, I thought that this was an interesting thing to try, and see whether it improved on flagellin. Was I getting most of the benefits of FMT by just doing a flagellin immunization, or were there further benefits to be had? And I think the data fairly compellingly shows that. So active microbial diversity is like, you know, it's a nonsense measure. It doesn't correlate with, you can say, okay, this correlates with health. Maybe it does, but that doesn't tell you, you know, why it correlates with health. It's just literally how many species do I have, some sort of measure of that. But obviously, if you do an FMT, you're going to have more diversity of species, but that doesn't tell you whether it was a good thing or not. The other markers that kept on going up are the more interesting ones. Such as digestive efficiency. So I think the answer is that that this tells us something interesting about what the major problems are. If you can do flagellin immunization and get, you know, major improvements in these metrics, and you don't get much more of a benefit in most of them by doing FMT, then that suggests that the presence of inflammatory inflammatory microbes is the major issue. But again, this is N equals 1. Somebody needs to run a big study, et cetera, if that answered your question.
Yes, I think I can see the trends, and I think that's quite exciting, even if it's just yourself. Yeah, I just want why at first, I mean, I guess you were not sick or you don't have any chronic condition that made you think that your microbiome is unhealthy to go to a fecal microbiome transplant.
This wasn't a, I wasn't in any way sick. I just, I was intrigued by the early changes in the microbiome. And generally my self-experimentation has tried to look at things where I would expect to see a signal. So this one was an easy experiment to organize, and therefore and one that might show a signal. So I rolled the dice, and actually it does show a signal. And I've used antibiotics across my lifespan about the same amount as any other human being. And as you can see, my before I did this, my gut microbiome is really not good. And after the flagellin immunization, really night and day on the mental clarity. There was an enormous change from just how it felt to think, which is interesting.
That's amazing. And then if I can, maybe I want to get your perspective on something which is, obviously we still don't know for many conditions if microbiome is a cause or consequence of it. And I believe we still don't have that answer for aging. And my question here is that we know that, for example, for the very first year of life, we have higher lactobacillus, bifidobacterium, because they help the baby to digest the milk and oligosaccharides. So I wonder, do you think that maybe the aged microbiome could also be because it helps an aged person to thrive?
Yeah, that's absolutely, that's absolutely possible. The components of it are adaptive rather than maladaptive. The fact that you can do this microbiome transplant in mice and get and killifish and get improved health suggests that those adaptive components are not as important as the maladaptive components. That would be the sort of thing I would look at here. But again, a lot more work is needed on this in order to understand what we should be doing in order to produce benefits. I think it's fairly clear that that putting a young microbiome in an old person is overall beneficial when a person is a mouse. For actual humans, yeah, we did a nice big clinical trial.
All right, thank you. Yeah, I agree with that. I would love to see some human data to see if indeed, it helps or there is some side effect of rejuvenating the microbiome somehow, because we're putting species that probably we don't even need at that age anymore, and we are removing something that we might need. But again, you're right, we just need more data, human data.
Okay, 10 minutes left. Any other questions?
I have a question about this. Yes, sorry, Evan. So the chart looks really good, and I'm curious, have you noticed any correlations with any other biomarkers you might be measuring not related to your microbiome?
No, nothing really changed. This was I saw a distinct absence of any changes beyond the microbiome changed. I felt a lot better, but none of the usual stuff that I measure was really much changed by that. So interesting. So in this case, cardiovascular, blood counts, all the rest of it, they didn't really do much.
Did you expect any of that to change, or is this?
No. You just got purely exploratory. You just do a set of simple things and see what should happen. There's some very obvious inflammatory markers you can measure, oxidative stress markers you can measure, and if you see something, great. If you don't, well, that also tells you something. So in this case, I saw exactly nothing other than feeling better.
So that that raises a good topic that I have struggled with is there any kind of marker that correlates well with feeling good or feeling bad? I spent a lot of time looking at and studying heart rate variability, and there was one study that said that correlated better with people's subjective feeling of health than any other biomarker, but I don't think that's the end of the story. Do you look at heart rate variability at all, and what are your thoughts, if so?
No, heart rate variability, all cardiovascular measures are so hard to measure, right? I mean, you have to do it the same position, the same time of day, the same diet, the same level of exercise, the same distance since your last exercise, and even then, it's all over the map. You have to take a 2-week-long baseline to have anything that looks reasonable. And I used to do a lot more cardiovascular measures than I do now because I found that they're basically useless if you're a fit sort of 50-something. You're just not going to get a signal out of anything we can do right now.
All right, thank you.
And if no other questions, happy to
I
wrap.
had a question. I actually had a few questions, but one was in regards to heart attack. Is there any relationship between bad gut microbiome and heart attack when you're older?
I'm not recalling one, but I'm pretty sure that if we went looking for one, we'd find it. Most of the research I'm aware of is focused on neurodegenerative conditions.
Interesting. And the other one was, is there any kind of relationship between the plastic pollution that we are exposed to and the microbiome situation in our...
Yeah, way too early to say. We don't even know for sure whether the microplastic situation is actually bad. I mean, there's just literally 2 or 3 studies where somebody is saying, yeah, this doesn't look good, but in all of those cases, there could be an explanation that has a lot more to do with I drink a lot of drinks with sugar in them from plastic bottles. The microplastic situation lacks the epidemiology that exists for particulate air pollution. So if it's very informative to compare the body of work for particulate air pollution, the epidemiology there, versus microplastics. We don't have the epidemiology yet for microplastics to actually even say that they are bad. I mean, it seems a reasonable supposition, but and people, since it's easy to act on less plastic in your food intake and drink intake, why not? But one has to also acknowledge that the data isn't there yet. It may turn out that microplastics are the worst thing ever, the cause of obesity, the cause of Alzheimer's disease, who knows? It may be, but we just don't have that evidence.
So even if it's like things like plaque buildup from plastic, that's something that we don't really know anything about yet.
We know nothing about that.
Oh, interesting.
Can I ask one last super quick question? It's just a curiosity, actually. What were the main challenges for the fecal microbiome transplant for you?
It was there were no challenges other than the fact that I just gave myself fibromyalgia. Everything else was very straightforward. If you don't have a donor, I can see that sourcing a donor would be annoying. But there's strategies. I mean, you can go post on bulletin boards in the local university, things like that. But if you don't have a donor around, it's sort of an awkward conversation to have with people.
All right, thank you so much.
Okay. In which case, happy to wrap here and appreciate the opportunity to chat to you, folks.